first project. First project that scientific project that I started since I come to Turkey some 6 years ago. So basically, what's going on? Why it's not working? Basically, what this group is really interesting for is adaptive radiation. And to give some background on adaptive radiation, that's a phenomenon when you have at some point in the past an ancestor of a group of species, and this common ancestor, starts to diverge, and all the different populations, they start adapting to a very wide range of habitats. So two famous examples are Darwin's Galapagos finches, so this group of birds which were first described by Charles Darwin in the Galapagos Islands. And so is on each island, and in is also many species that living within on each island as well. So they were able to occupy a very big number of ecological niches. Some of them are specialized of eating seeds, some of them are specialized on eating insects, and so on. Another famous example is this Anolis lizards in the Caribbean, which also a similar situation. At some point in the past, a group of ancestral lizards arrived from the mainland, South or Central America, and they diversified all around these Caribbean islands, also getting into all these very different ecological niches. Some of them living very high in the trees, some of them live on the ground, some of them are very large, some of them are small, all the different shapes and colors and ability to live and thrive in different habitats. So as you can imagine, and if you watch any sort of BBC Nature documentaries, most of this adaptive radiation, this big groups of animals or plants with a big number of species, they all come from the tropical areas. So that's where the most biological diversity is found. However, so there are examples from the non-tropical areas. So from the Palearctic, we have this group of lizards, Darevskia, which is a group of lacertid lizards. So it's a big family of lizards which are found in mostly in Africa, Eurasia, not really so much in anywhere else, not in America. And so they live in Palearctic region. So they, this habitat has winter, and nevertheless, because of the, all the some unique peculiarities, and because of the probably mountain landscape in the Caucasus, they diversified quite a lot. So there are currently about 25, maybe more, described species of Darevskia, and they also come in different shapes and colors, and they all, many of them adapted to different habitats. So as an example, there's a contrast between these two species. So one of them is this Darevskia rade, which is a rock cliff specialist. So this is a lizard. If you have been to like Van or any other sort of southeast Anatolian areas, not Chernak, but a bit higher in the mountains, you would see them on the walls, on the rocks. So they are really specialized in living on almost vertical rock cliffs. And as you can see, this lizard has a very sort of narrow head. So it allows it to squeeze into all the different crevices and cracks on the rocks. And that's the opposite example. This is Darevskia derugini. So that's a species that lives basically on the ground in the forest. It almost never sort of climbs rocks. If it does, it doesn't really stay there. It hides on the ground, and because of that, it actually it doesn't need to squeeze itself into the crevices. So its head is more round, which helps with hunting insects, and so this species has a stronger bite force probably. And as well, so the other species are found from in different altitudes, in forest, in coastal habitats, in the mountain habitats, and so on. It will be too much to. I don't have really much time to go through for every species now. So another unique fact about this group of lizards is that there are 6 parthenogenetic forms. So let's call them species for now. So parthenogenesis is a biological term. It means virgin birth. So basically, that's ability to develop without fertilization. So the females, in case of the lizards, they lay eggs which are not being fertilized, and then diploid females are able to produce diploid offspring, and all of this offspring is also females. So all of these parthenogenetic forms are found in nature, and there are currently 6 of them. Well, there were 7 described species. 2 species are actually the same, so they should be like confined into the single taxon, and all of them are found here in this region, which borders Turkey, Armenia, and Georgia, right? So it's really compact area in between Lake Van and, let's say, South Caucasus, going a little bit into Azerbaijan, and we will get to the possible reasons why the distribution is so compact. Sorry, yeah. So before we get into Darevskia, I have to digress a little bit, and I think I'll need to tell you a little bit about the like general theory of asexual versus sexual reproduction in animals as well as in plants. So asexual reproduction is it comes also in different shapes. So you can think about plants, of course, which yes, so many of them are able to reproduce vegetatively. So if you cut a small bit of potato, a whole potato plant will grow from it. And then animals have various different mechanisms. Some of them are very strange, peculiar. So like this gynogenesis, where they require the sperm of a different species to be present in order to develop without fertilization. Right, and then there is like a true parthenogenesis, where, like in case of the Darevskia lizards, where females can just reproduce without any males or any other species being present around. So across the biological kingdoms, of course, plants are able to reproduce asexually routinely. Then parthenogenesis, asexual reproduction, is found relatively often in many invertebrates. But it's not really that common in vertebrates, right? So again, fish, you can find asexual reproduction in some fish and some amphibians, like the salamanders or sharks. You can have the gynogenesis in some fishes, and interestingly, quite a big number of reptiles, quite a big, not just all reptiles, but specifically squamates, so snakes and lizards, they have parthenogenetic reproduction, and even birds are known to reproduce parthenogenetically, but it's not really, well, it's not that common. I think there are just confirmed examples in, I think, Turkey species and the California condor, but they never, this the offspring is not really able to survive for very long, but it's still possible. There is no asexual, there are no asexual mammals at all. So probably because well, we will get into discussion as to why it's not probably possible. So let's see, like from the evolutionary perspective, which are the benefits and which are the costs and disadvantages of having sexual reproduction versus asexual. Of course, if you think about asexual way of proliferating, well, that's actually quite effective, because every individual is able to produce 2 or more individuals, and there is no wastage of time, no wastage of resources, no need to finding a mate, and so on. So think about this. One species of giant tortoise from Galapagos, from, I think, Piña Island. So his name was Lonesome George. He was a male. It doesn't matter if that was a female, it would be the same. It was the last remaining individual from the entire species. So there was no mate it could possibly reproduce with, so it died a few years ago. And then, of course, think about some plants in the tropics which can only be pollinated by a very specific species of bat or some moss. And so if this bat is going extinct, then the plant is also probably being extinct very soon. So sexual reproduction is very costly and dangerous for that reasons. So, however, we need to think about the possibility of the opportunity which sexual reproduction provides. And that is a combination. So you are really able to sort of find all the different combinations of different genetic elements, different genes, and some of these combinations Well, first of all, they're going to be deleterious. So if you can imagine that, all the phenotype of an individual can be sort of reduced to one dimension here. So let's call it just a possibility or probability of survival probably based on 100 different traits, but let's just collapse everything into one dimension. So this is survival. This is a probability that this particular organism will live and produce more offspring. And so some of the combinations will be very bad. So they will, lots of bad genes, lots of deleterious elements, and so they will be selected against. So this left tail of the distribution is not going to survive. But at the same time, there would also be very successful, evolutionarily successful combinations of genes, genotypes that will be favored by selection. And so this small right tail of the distribution will actually be able to sire the whole new populations in the future, right? And of course, in the clonal or asexual reproduction, everybody is more or less like the other. So think about this apples or any other commercial crop or commercial plant. So these apples are very nice and they are sweet and they are very, they are really selected for, very favored by the humans. But they're all the same, right? So if somebody doesn't like sweet apples, well, then their fate is doomed. At the same time, if the same species, apples which are reproducing by seeds. So if you reproduce apples as seeds, most of them will be very sour and bitter, not very edible. But you can also come up with some very nice varieties, and so, they might become a new breed of apples and also very successful. Okay, so asexual reproduction, on the other hand, is also very useful when you don't care about evolving too much, but what you care about is to quickly colonize and quickly occupy a new habitat. So in the ocean, a lot of sedentary or semi-sedentary organisms are able to reproduce asexually very quickly, and also the plants, of course, the potatoes can proliferate without meiosis, without recombination quite a lot. Right. So some species have found an optimal solution. They are able to combine both asexual and sexual reproduction. So aphids, for example, they have the cyclical parthenogenesis, where most of the time they are reproducing without males parthenogenetically, but then occasionally, not occasionally, but in the end of the season, they produce sexual forms. And so there is recombination, so they are probably making sure that the whole species is evolving, but then just to maintain a very big number, very large population, the parthenogenetic forms really thrive in the summer. It's a very similar situation in the water fleas in Daphnia, and in many other forms as well. Even those very interesting organisms, such as Bdelloid rotifers, which are tiny microscopic relatives of roundworms. So some of them were thought to be asexual, and they thought to have been living without males for around 50 million years. So that was a bit surprising, because people thought that, well, that species cannot survive for such a long time because it lacks genetic recombination, it lacks variability, it cannot evolve. And but nobody could really find any males in this species. They still didn't find males, but they found genetic evidence for sex and recombination in this species. So, once a million generation, or maybe once every 1,000 generations, there is a male in these tiny animals, and that what probably saves them from the evolutionary, doom fate. So. What we're going to be talking about here is parthenogenesis by hybridization. So there are two ways. You can imagine that occasionally an organism would just produce an offspring without fertilization. But in many successful cases of parthenogenesis in nature, what you have is that you have hybrids which start reproducing without males. And that's quite common in the salamanders and fish, and of course in lizards, which I showed you, and in all of the really successful examples of parthenogenesis in reptiles, in snakes and lizards, all of them have a hybrid origin. So this geckos in Australia, this teat lizards in North America, and Darevskia, so they are hybrids between two different bisexual species. And so in case of, I will get to it later. So in case of Darevskia, yeah, probably I'll have to here. So we have these two groups of parental species that also live in the same area in the Caucasus. And as you can see, so this species, Darevskia valentini here, it sort of occupies a quite large area, stretching all the way to Taurus Mountains. But then in this eastern Anatolia, its range overlaps with Darevskia radi, right? And then there's two other groups, Darevskia porchinski and Darevskia mixta, and also valentini and mixta. So their ranges overlap here in southern Georgia and Armenia, right? And this is exactly where the parthenogenetic hybrids are found, right? So even if you look when you're looking at the geography, of something, you can intuitively understand that this hybrid species must have originated quite, somewhere locally. And of course, they have lots of morphological traits, which are somewhere intermediate between one parent and the other. Right. About 22 years ago, people already looked into the sort of genetics of these groups using allozymes, so the proteins with different electrophoretic charge. Electrophoretic properties, and they really found out that here they compared the alleles or electromorphs of different proteins in the bisexual species and in the hybrids, and they really found that, for example, here all these hybrid species, they are heterozygous for this CH alleles, right? And one H allele is coming. I, for example, I is coming from the male of Darevskia porchinski, that's a paternal species, and C comes from Radi. Yeah, so this is another bisexual species, and so on. So just by looking at the genotypes, you can see that most of them are heterozygous for the forms inherited from the specific species of specific bisexual species. So they and then people also used mitochondrial DNA to identify the maternal origin of the species. So more or less, it was clear the parental origin, the identity of the parents for each parthenogenetic species became more or less clear even 20 years ago. But of course, this is very broad, and it's possible to get this same picture in much more details, which we try to do. All right. So what we have done since this project started is in collaboration with my very good colleague in Tbilisi, in Georgia, David Tartnishvili. So we decided to kind of look back into the details of the parental origin of all these 6 parthenogenetic species, right? Because if you think about this scheme here, well, it's very general. For example, Darevskia valentini, here it's just listed as a single species, but of course, as you can see on not here, as you can see on this map, the range of Darevskia valentini is very, very large. It stretches all the way from south of Lake Van to Georgia and all the way to Anatolia. So it's not really clear which particular population of this valentini has really given rise to the parthenogenetic hybrids. So what we did is, at the first stage of this project, we genotyped several hundred of samples, several hundred of lizards from all the species, all parthenogenetic species, and these 4 bisexual species. We genotyped them at a number of macrosatellite markers, and just to give you very, very sort of condensed information here, that we run as a few standard programs to figure out the possible origin of different alleles at these microsatellite markers. And it was generally it was really like, you know, the result confirmed the previous findings that were based on the allozymes. So here you have these two species, Darevskia dali and Armeniaca. Both of them resulted from the maternal parent Darevskia mixta. Here you have this group of endemic Turkish species around Lake Van. You can see that none of them not really different from each other, but also they're coming from just one pair of parents, Darevskia Rade and Valentini, and so on. So you can kind of not only see the hybrid origin of every parthenogenetic group, but also you can see that some of them are more similar to each other than the other. And what was really striking in this study is that although we thought that, for example, Darevskia Armeniaca and Darevskia dali have independent origin, so we thought that at some point in time, their parents, Mixta and Valentini, hybridized, and then there was Armeniaca, and then at a different point in time, they hybridized again, and there was Darevskia dali, right? But what the results of this microsatellite genotyping didn't really support this hypothesis too much, because what we see is that we have the same heterozygous genotypes in both of these hybrid species. Here you have Dali and Armeniaca, and here they have exactly the same genotypes at this locus, and exactly the same genotypes at this locus. And it's really difficult to imagine how it might be possible if the hybridizations were independent, right? So if it happened independently, then there is no reason why the same heterozygote genotype would come up in both of the species. So in the same situation, to slightly lesser extent is happening in other species as well. But of course, in these two groups, Bendimachensis and Sapphirina, they were almost the same in almost every marker. But this is just because they are really the same group, so they have the same common origin. But with Dali Armeniaca, you know, they have clearly different origin, and it wasn't really clear why would some, not all, but some of the microsatellite genotypes be the same in these two groups. So we thought about different explanations, and one of them is that it might be possible that the actual origin of these two species, Armeniaca and Dali, is much more complicated than we thought. So it's not just two independent hybridizations, but so what David envisaged, that probably there was a initial hybridization that gave rise to Darevskia dali, and then this dali hybridized again with Darevskia Valentini with a bisexual parent, and then somehow there might be some triploids involved, and maybe there was a ploid reduction back to diploids, and then you would have Darevskia Armeniaca. So such a scheme would explain why we see the same genotypes in Armeniaca and Dali. But of course, we don't really know the cytological mechanism that would allow this to happen. So we don't know why would triploid and how would this possible triploid be reduced to back to diploids and become Armeniaca. So this is still an open question. And so what we did in the second stage of this project is to genotype more or less the same samples again, but this time using restriction site associated DNA markers. This are rat sequencing, which has become very popular for this sort of molecular ecology studies, and it's based on the fact that there are restriction sites, so sites in the genome which are recognized by the restriction enzymes, and they of course they are found in more or less the same positions in different groups, even in different species, right? And so if you are sequencing those fragments of DNA which follow or flank these restriction sites, that you're very likely to get the same homologous loci, the same homologous fragments in different organisms, and then you can genotype them. So then you can figure out which alleles are present at a specific locus, and so on. And that's a very powerful method, which of course allows to get data from potentially thousands and thousands of genetic loci as compared to the handful of markers when you use microsatellites, for example. So Yeah, so basically, in the end, you are getting these SNPs, and then you're using this SNP data for again, to study the population structure and to study the relationships between different species or populations. As we were, so all of this were done in between 2017 and 2019, and then in 2019 we were, I think, extremely lucky, because what we thought at the beginning is that these Darevskia lizards are not really very famous model organism, and so there is no whole genome, no full genome sequenced for these lizards. If you think about full genomes, at least if you thought about full genomes 10 years ago, mostly these were human, domestic animals, plants, some important objects, mouse, maybe Drosophila. However, the number of whole genomes sequenced really exploded in the recent years. So in 2019, people in collaboration in Italy and America, they released the full reference genome assembly of another lizard, which is evolutionary pretty far away from Lacertids. So it's a wall lizard, Podarcis moralis. So it's supposed to be maybe there's like 20-25 millions of years of divergence between the Darevskia and Podarcis. However, nevertheless, because the chromosomes in all Lacertid lizards are very well conserved, so all of these Lacertids, the whole family, all of them have 18 chromosomes plus chromosome Z. So when we tried and we aligned our sequences from Darevskia to the Podarcis genome, we get at least 50% successful alignment. So that's a very high percentage. And suddenly, even we didn't expect this at the beginning, suddenly we were able to actually resolve the genotypes of our samples at chromosome level. So we can look at each autosome, and we can also look at the Z chromosome there. So that was quite a big help. And so immediately what we thought about is that, well, the following situation. So Darevskia, as well as some butterflies, some birds, snakes, and so on, they have female heterogametic sex. So their chromosomes are labeled Z or ZZ or ZW. So the only difference between ZZ and ZW is that when your male sex is heterogametic, you call it XY, and when the female are heterogametic, they call it ZW. So what does it mean? Males have two identical chromosomes, ZZ, unlike mammals. Females have just one big chromosome, Z, and a tiny chromosome W, which maybe contains some genes, but it's very small. So it's analogous to Y chromosome in the mammals. So think about how the parthenogenetic species have originated. So they are all females. So all of them are ZW, and their W chromosome obviously comes from the females. So it's no big surprise. But again, their Z chromosome can only have come from the father, right? So even though in bisexual species, Z chromosome can recombine, maybe not as often as like W doesn't recombine at all, and not as often as autosomes, but in the bisexual population, the Z chromosome will still be recombining and exchanging genes. In the parthenogenetic species, there is just single Z chromosome. It doesn't have anything to recombine with. Anyway, there is no recombination. It's a clonal population. So this Z chromosome in the parthenogenetic species is kind of preserved exact copy of its paternal Z chromosome, right? It might accumulate some mutation along the way, but it will still be just one big intact sequence. So in a way, it's analogous to the mitochondrial DNA that, as we know, is only inherited on the maternal side. So we were very excited about the possibility of comparing the Z chromosome in the hybrids, in the parthenogenetic species, and in their bisexual parents. And why would we do it? Is that because, as I mentioned before, the range of the parental species, the range of Darevskia valentini, a father, species and Darevskia radae and Darevskia mixta. So they're quite large, and we don't really know which particular populations of the paternal species was the real father. Of course, it's not the extant population. Those animals, those lizards which we can see now, they're not exactly the exact same population which was the father, because hybridization happened some, potentially hundreds of thousands of years ago. But we can still ask a question: which extant population is genetically closest to the real father, right? So again, this is the same sampling effort here. So we had more or less the same populations all around Eastern Anatolia, Georgia, and Armenia. And so what we were able to get is when we only looked at the chromosome Z. Again, we just, because that doesn't recombine in the hybrids, we were able to build very robust phylogeny based on the sequence. And as you can see here, we have like some sort of outgroup. So these are maternal species. They also have Z chromosome, of course. So the Z chromosome is very far, and it has no relation to the hybrids. So there is no connection between the maternal species and the hybrids, which you can see here, based on Z chromosome, which proves our well, kind of obvious assumption that Z chromosome is not inherited from the maternal side, right? And then, as you can see here, every one of these hybrid species, every parthenogenetic species, is clustered very nicely with the specific population of the paternal parent. And it's we can actually look at it as very sort of fine details here, right? So, for example, here we have this hybrid parthenogenetic Darevskia Rostombekovi, and it clusters nicely with its father. So we know that it's Darevskia Parchinsky is a father species of Rostombekovi, but it's not just any Parchinsky; it's a Parchinsky collected in Armenia, and Rostombekovi is also from Armenia. And then we have another Parchinsky population from South Georgia, and it clusters very nicely with its another daughter species, Dali, which is also found in Georgia. So this is Georgia here. And then we have, very surprisingly, we have another group. So this is Darevskia Valentini, again, which is split in different populations across its range, even across Eastern Anatolia. And so one group of Valentini, which is found in Georgia and in northeast Anatolia, so it's area around Ardahan and Kars, and also in South Georgia. So that appears to be the father of a species Darevskia uzeli, which is again endemic to Turkey, but it lives slightly to the north. And then it also appears to be another population from Georgia, appears to be the father of Darevskia armeniaca. And if you look geographically, let's look at the geography now. Sorry. And geographically, here we have armeniaca, and in the same place we have Valentini here. Here we have uzeli, and in the same place we have uzeli. We have Valentini from Ardahan, and so on. Here we have Parchinsky Armenia and Rostombekovi, Parchinsky Georgia, Dali, and also going all the way to like even Erzurum. So this is Erzurum, population of Valentini from Erzurum. It appears to be the father of this parthenogenetic species, Darevskia unisexualis, right? So geographic distance here, well, is not very small, but it's still the nearest population. And in Lake Van, it is the same. We have Darevskia Valentini population, which we collected near Lake Van, and it's clusters together with Darevskia sapphirina and Bendimachensis in the same area in Lake Van. So this is really for the first time we were actually able to look at these fine geographic details of the paternal origin of parthenogenetic species, right? As before which people just could say very generally, okay, it's Valentini, which is the father of all these groups, but which Valentini, it wasn't possible to tell. So yeah, so we can conclude from this results is that hybridization, which gave origin to parthenogenesis, was really very much local event. So everything happened like as short as possible. So probably neither the parental species nor the parthenogenetic species have moved very far from the place where the hybridization took place. We can also say that from the phylogeny that there were two time periods at which hybridization event took place. So one of them is let's say, so around here, all these 4 species here. So that would be, and also here, it will be Darevskia Porchinsky, Darevskia Dali, Uzeli, and Armeniaka. So they all kind of split from the nearest paternal population around the same time. We have some estimates, so it may be maybe it's 500,000 years. We don't really trust these results too much because the molecular clock calibration really depends on which which known split time you kind of enter into the model, and so it's known to be unreliable. And to be honest, 500,000 years is still a very long time. But relatively, so if we forget about the absolute numbers, relatively, this time period is definitely more recent than this time period here. So here we have the origin of Darevskia unisexualis and Darevskia sapphirina benimbachiensis, which are now species. So that happened, much earlier in time. Again, we can argue about the absolute age in the millions of years here. And that also makes sense geographically, because the first group here is found in this area, kind of to the north, South Georgia, and northeastern part of Anatolia, eastern Anatolia, so Ardahan, and this area, like Van and Erzurum, are a little bit to the east and to the south, right? And geographically, there's even like some nice sort of mountain barrier between the two. So we can also, envisage that there were some geographic reasons for this hybridization to take place at different times in different geographic areas. Right. So this is an updated sort of reticulate network, reticulate phylogeny of the parthenogenetic species, and now we can just very reliably say that okay, these 2 species, Armeniaka and Uzeli, they have come from population of Valentini, which is now found in Georgia and northeast Turkey. Porchinsky from Georgia was the father of Dali. Porchinsky from Armenia was the father of Rostambekovi, and then we have Valentini, 2 different populations of Valentini in Erzurum and Laikvan, and respectively, they seem to be the closest bisexual ancestors of the species Unisexualis and Sapphirina benimbachiensis. Right. So I think, I don't know if we still have time, but if I'm yeah, I guess we can leave more time for questions. I just have to, I would like to acknowledge the participation of a lot of my colleagues, other people in this work. So first of all, to my colleague and scientific sort of partner in this grant funding, David Takhnishvili from Ilia State University in Tbilisi in Georgia. And so we started the field work in Eastern Anatolia with Mehmet Kürşat Şahin, who is now in Karaman, but hopefully he'll move to Hacettepe soon. So he was really instrumental in doing the field work. And then Meriç Erdoğlu from METU, from Middle East Technical University, who is now almost finishing his PhD, which is all about, again, Darevskia lizards, but those populations which are found around Lake Van. So I didn't speak too much about this work, because I think maybe next year you will have opportunity to listen to his results at some occasion. So we really like to thank all the people who provided samples and also helped with the field work. And this work was funded by the bilateral program between TÜBİTAK and Shota Rustaveli Foundation. So it was a bilateral project between Georgia and Turkey. Very well. So I think that's all from me, and hopefully I can answer questions should you have any. Thank you.
Thank you, Alexey. This was very interesting. I mean, the model and all the species involved are very interesting, and your results are very interesting along the same lines. Are there any questions that any of the audience would like to share? Let me check if there's anything in the chat as well. Okay, I don't. Yeah, maybe the topic is like a bit more like a topic that a lot of our students are not accustomed to at some level. But like I'd like to ask a few questions then, just maybe to start the discussion. I was wondering like your thoughts from a taxonomic point. Like I'm not sure if there's like a clear answer or like a straightforward answer to it either, but I was thinking like the Valentini that are the parental species, they are distributed all along the phylogeny that you showed us at the end. And I was wondering like, at what point might we want to call those different Valentini different species themselves rather than referring to all of those as Valentini?
Of course, yeah. So that's really, let me go back to this. Yeah, so the phylogeny, for example. Absolutely, yeah. So it's an ongoing debate. Yeah. So it's not a single species, most likely, because it has such a long, such a wide range. The additional problem that all of this group has, so all of the paternal bisexual species, I hope you can see my screen now. So all of them, this Porchinsky and Valentini, and also other species here. So there's like Darevskia rudis, which is not included in the origin of parthenogenetic species, but it's still there. So they hybridize between themselves. Right, so there is a strong gene flow between, let's say, Valentini and Porchinsky. So that's a case of like when you can't really define, or you should be defining species based on, the ecology or genetic origin or evolutionary like status, but there is gene flow and there was gene flow between them, and that's another sort of interesting study because despite the strong gene flow, they are still different ecological types. Right, so Darevskia Valentini is a mountain specialist. It's found in the higher altitude, let's say above 1,700 meters. And let's say Darevskia Porchinsky is a very different lizard. It's found in the forested areas on much lower altitude. And then even among Valentini, it's not really clear what people should really be calling Valentini because so there are different groups. And all of them kind of have the similar ecology, so they're all mountain species. But then, for example, in Taurus mountains, in Bolkar Mountains, actually in Nidea, so there's a population which used to be called Darevskia Valentini because it's found high in the mountains, but it's actually Darevskia rudis genetically, right? So it has the same ecological type as Valentini, but it's not Valentini. So it's quite a big mess, and what we are dealing with is that I'm using the species names, especially on this paternal side, but in reality, maybe I should be calling them like OTUs or taxa and specific taxons, because that's a classical story of when morphological traits and ecological traits don't always correspond to the genetics. So. Yeah, so it's complicated, I guess that's an answer.
Yeah, so are like if you just looked at them morphologically, like are the Valentini from Ardahan, Georgia, Erzurum, Lake Van, Children, are they pretty much the same? Or can you tell?
No, they're not the same. They are different. So again, different herpetologists who used morphological traits, they still describe them as one species. But probably they were thinking about that's the fact that there is no other group representative of the same group. So the closest one would be Darevskia rudis, which is found on the Black Sea coast here. So and then it's on much lower altitude, and so yeah, it's really, it depends on your interpretation of the importance of like different morphological traits. So these two are different from each other. They are more different than they are more similar than Porchinsky, for example, because these are two big lizards and we live in the mountains. But it's also different from rudis but then you can also find some morphological traits like folidosis, number of scales in some parts of the body and so on, which resemble Rudis and resemble Valentini. So yeah, without genetics, it's really, really, really difficult to understand which is which. And
yeah, very cool. You also like refrained from referring to a specific timeline for splits, like when different groups split from each other. Yeah. But I was wondering, like, just thinking of the formation of the parthenogenetic species from like the sexual parent, like how long does that theoretically or based on empirical evidence take? Like, is that like within the ballpark of like hundreds of thousands of years, or more like millions of years?
Yeah, well, it's
or can it be a very quick process to like?
Again, there are more questions than answers here. So, so this is molecular clock data, and it's based on some calibration points. And we use two calibration points. One of them is older, one of them is more younger. That's why we have two datings here next to each node in the tree, right? So, but again, so both of these calibration points, they're based on some different studies. But those studies have, quite good data. So one of them is like genome-wide, I think, like transcriptome-based genotyping. So it's a good chunk of nuclear DNA data. Another is based on full mitochondrial genome. So you should be like trusting this. But on the other hand, they are like really, a bit. So let's say they're much earlier than anybody have thought before. So initially, when these parthenogenetic species were discovered, like 40, 50 years ago, people thought that, well, it must be like 10 or maybe 15,000 years the most, because they think, okay, that must be something that happened just after the last glaciation. So because at the last glacial maximum, all of the Carcer room plateau, all these high mountains, it was not really suitable for lizards. It was very cold there. And so no, probably there were no lizards there, and then suddenly they appeared. So okay, so they thought maybe hybridization took place right after the last glacial maximum. But well, that's a bit too early. But so now we can see it's 500,000 years. There was, yeah, so it's hard to say. There is another group of parthenogenetic hybrid lizards in North America, and there was a recent study where they used much better data than ours. So it was like, I think it was also rat sequencing, but much denser genotyping. So they had enough data to run not molecular clock, but they could run a sort of coalescence-based model based on demography. So they looked at the the possible common coalescence point of origin of the population, and they estimates where they kind of into the maximum were around 500,000 years. So 500,000 years for these North American lizards is kind of similar to 500,000 years for this sort of 4 species that we have. Okay, but in our case it's a lower, well, it's a median, but it's the confidence limits are very small here. So they kind of overlap. So you can kind of imagine that for lizards, at least for American lizards, another group, it might be possible to live that long. But yeah, still it's we probably need more data. So I would really like to work with like bigger parts of genome, and hopefully be able to run this demographic modeling, because that seems to be now more reliable methods compared to molecular clock.
Oh, thank you. Yeah, it seems not necessarily like an example of rapid evolution, but more like the classical example of vicariant evolution combined with the kind of special like sexual / asexual mode of reproduction.
Also the fact that they are very local. As I say, like in Lake One, for example, there's a population of Valentini and population of a hybrid Bülent, and they all like 2 or 3 kilometers apart. And so it's very hard to imagine that this kind of situation could persist for such a long time, like 1 million years, and nothing happened. There were no range shifts. There were no like major migrations. Well, as we know that this area was a scene for glaciations. well, there were lots of climate changes in climate, lots of events in this area. So it looks really interesting if these lizards were able to survive in the same place for such a long time. So yeah, molecular clock seems not to be not so good for at this scale at least. Yeah.
Okay, thank you. And I have a final question, because we are an Institute of Environmental Science and interested in like different environmental issues, including the like loss of biodiversity being a big problem. Could you comment on the conservation implications of your results?
Yeah. It's an important thing. So so as I was talking before, the many parthenogenetic species actually appear to be very successful, right? So and it's also the case for Darevskia, but not for all of them. So let me share this again. So if we let's go back to so these two species, for example, Armeniaca and Dali, that are found in South Georgia and northeast Turkey near Ardahan. So they are really abundant, right? So you can find places, especially Dali, it lives in the forested area. So this is very common basic species in southern Georgia, and it's everywhere really. Then there is a species Darevskia unisexualis, which is part of its range in Armenia and part of its range is in Turkey. So it's around again Kars, around Kars and going all the way to Armenia. That's also very abundant species. If you go to Kars Kalisi, so the main tourist attractions, it is everywhere. And it makes sense because they can reproduce very quickly. They don't waste any resources on males, and so they are really prolific there. So all in all, this group of species here, they all seem to like confirm to the fact that parthenogenetic species, at least locally, are able to survive very well. There is even study that shows that Darevskia Dali in Georgia is kind of able to outcompete its parental species. So Darevskia Dali found somewhere on like in the medium altitude between Porchinsky and Mixta. So Mixta is on the lower altitude, Porchinsky is slightly higher, and so Dali seems to be occupying the middle area, and it's, even seem to be slightly like driving the parental populations out. So that's very successful. So even though they might be successful locally, obviously, but the theory predicts that they might not be so like responsive to the changes in the environment. So big changes, climate change, especially for the soul species that live on higher altitude. The theory predicts that they might not do very well because they have much weaker potential to adapt to the changes. At least it's a theoretical prediction here. However, for the moment, this group of species seem to be doing well, but they are very endemic. Uzeli is found on very small area here, right? So it's like a really endemic species. Unisexualis has slightly wider range, but still not very long. Dalia armeniaca again, so they're still endemic. So locally they are abundant, but there's this danger of extinction because of endemism. However, we have this another Turkish endemic, which was thought to be two different species. It's one thing, one population, Sapphirina and Bendimachiensis. So these are neither locally abundant, nor they are nor there are like very many populations. In fact, there are only 4 known populations of this sort of hybrid species, all around Lake Van here. So actually, Miric had a small map here. Yeah, so here is a map. So here's only several populations. These are parental populations here. So here they are, just these several populations of Bendimachiensis and Sapphirina. And in just one population, they seem to be locally abundant, so on, but otherwise they are not very, the density there is not very high. And one of these populations is literally on the side of the highway. and they this open mining. They just mining the, I think, like stones from the face of the rock there. So they are gradually eating away the area, the only area where these leeches are found. Here is this area, by the way. So yeah, so here's Mehmet, Kürşat, and Meriç. So this is like the only place where one of the only 4 places where this species is found. As you can see, there is nothing there, and next to this slope there is a like open mine. So so for some reason these 2 species, Bendimiasis and Sapphirina, they don't seem to be successful, and they are clearly on the decline. So that's very sad. But some of them might sort of survive a little longer. That's what I can say.
Okay, thank you, Alexey Hoca. Are there any other questions from the audience? Okay. Okay, thank you very much, Alexey Hoca, and yeah, it was a very interesting talk for a very special group of species. So yeah, thanks for sharing your like research with us. Okay, Pınar Hoca, do you have anything to add?
Thank you very much for attending the talk, and thank you very much for the excellent, exciting talk. Thank you, Alexey Hoca.
Thank you for this opportunity. Very well.
Okay. Bye, everyone.
Bye-bye.